Why, hello there. Let's go through the neurotransmitters today. Neurotransmitters made in the synapse bind to the receptors on the postsynaptic cell or the terminal, and they are neuromodulators, and they only affect the strength of the synaptic transmission. So they influence the synthesis, release, degradation, and uptake of the neurotransmitter. And these are the main ones. The vesicles are made in the presynaptic body and then shuttled by fast or slow transport to the nerve terminal. The enzymes needed to make this small molecule transmitters are made in the rough endoplasmic reticulum, transferred to the Golgi apparatus where they are modified, and now modified, they are soluble and they move down the axon by slow axonal transport via the microtubules. Most neurotransmitters are made in the clear core vesicles where they are recycled. So these are the clear synaptic, goes down, slow axonal transport, and the fast vesicle recycling occurs. Large, dense core receptors are going to make the polypeptides in the rough endoplasmic reticulum. Opposite, it's going to have a fast axonal transport down to the terminal, and that's where the propeptides are broken up, and there are no recycling of the neurotransmitter, and that's because they have further to go, right? They have, if you can go back to this picture, they have a long distance in comparison to the short distance. These are going to be your neuropeptides. They're going to go to the dendrites, and they are going to work via a G protein coupled receptor. Well, in the clear synaptic vesicles, it's going to use the ionotropic receptor, so it's going to be faster, and it only needs to go across the synapse junction. So the large dense core vesicles will also make serotonin and noradrenaline or norepinephrine, and it needs calcium to release the vesicles. And each vesicle releases a single quantum of neurotransmitter. What does that mean? It's basically like a pre-packaged amount. So here in the presynaptic cell, these are going to be your dense core vesicles, and here the clear vesicles are going to predominate. And there you go. Here's the mitochondria, the powerhouse of the cell. Here is the synapse. So clear vesicles, they're just gonna just shoot across the synapse. And here's the postsynaptic cell. Okay, so calcium comes in thanks to the action potential, and the vesicles are going to be exocytosed. The snare protein will help the proteins dock onto the presynaptic membrane, and then the contents are emptied into the synaptic cleft. The excitatory receptors, NMDA, are going to increase the depolarization and firing rate. The inhibitory receptor, GABA, will hyperpolarize the membrane and decrease the spike frequency. So, the synaptic vesicle, here you go. Well, these are not things that you need to know in a lot of detail, but synaptobrevin is going to be on the vesicle, and that needs to bind to the SNAP proteins. And they have to prime and fuse in order for the synaptic vesicle to release its contents. Ligand-gated ion channels are going to be your nicotinic acetylcholine receptors. G protein receptors are going to be your muscarinic. Kinase linked is going to be your cytokines receptor, and the nuclear receptors, these are your hormone receptors, and this is the amount of times it takes for the action to occur. Termination due to enzymes. It's going to stop the action potential by degrading the neurotransmitter. The most important ones are acetylcholinesterase and monoamine oxidase, or MAO. The nerves and support cells will reuptake neurotransmitter and repackage it in the synaptic vesicles to save on energy. It takes a lot of effort to make the neurotransmitter, and it also takes a lot of time. So in poisons, particularly like black widow poisoning or organophosphate poisoning, it uses up all of the neurotransmitter, and the body just can't make enough, especially acetylcholine, right? Acetylcholine is used for somatic muscle contraction, and without the acetylcholine, the diaphragm isn't able to contract, so person actually dies from respiratory failure. The diffusion occurs when the transmitter diffuse out into the systemic circulation, and those will be terminated by specific enzymes, or they are going to be excreted by kidneys. Okay, so acetylcholine is made from choline and acetyl coenzyme A, broken down in the cleft by acetylcholinesterase. Choline is going to be transported back into the axon terminal and is used to make more acetylcholine. All the preganglionic synapses use it, and the enzyme catalyzing the acetylcholine coenzyme A kind of process is acetylcholine and these will bind to your nicotinic and muscarinic receptors, and they work via simple diffusion. Okay, so the nicotinic receptors are going to be in the CNS and the adrenal medulla to release adrenaline or epinephrine. The nicotinic receptor is also in the autonomic ganglion and in the muscle for contraction. And the muscarinic receptors, M1, is going to be in the CNS and the salivary glands, parietal cells, and it will increase IP3, locomotor activity, and secretions. M2 is important to know for a pharmaceutical standpoint because it's in the heart. Particularly, this is going to be focused on like the conduction system. So it decreases CAMP, which decreases the force, the rate of the heart rates, and the AV conduction. M3 is the receptors in the smooth muscles. Those will increase IP3 and increase vasodilation and glandular secretion. M2, M4, and M5 in the CNS are inhibitory. So important to know. Okay, so once again, all presynaptic preganglionic synapses will use the acetylcholine as the neurotransmitter, and it's dependent on calcium release. Postsynaptic, for the receptors that use it, are going to be dependent on sodium for the depolarization. MACH receptors are going to be blocked by atropine, and rivastigmine is an acetylcholine esterase inhibitor, and that's a treatment to treat Alzheimer because it's going to allow the acetylcholine to stay a little bit longer in the synapses. Alzheimer reduces all acetylcholine-like amounts in the subcortical area and reduce the amount of choline acetyltransferase. So that's problematic, and that's how it causes the muscle weakness, the inability to control somatic functions. Okay, so venom. Corbotoxin and coral snake venoms will bind to the nicotinic receptors, causes flaccid paralysis, and like I said, death by respiratory failure. And you treat it by giving physostigmine or neostigmine to inhibit the acetylcholinesterase. So we don't want the acetylcholine to be taken up. We want it to remain in the neuron, in the neuromuscular junctions, to continue breathing. Okay, so it's also used in anesthesia. So two important compounds, succinylmethanium chloride and decamethanium, will bind to the receptors for an extended period of time, and it keeps the ion channel open by overstimulating both nicotinic and muscarinic receptors. These receptors remain depolarized, and they are not responsive to the normal acetylcholine esterase release. So this also forms like flaccid paralysis. Okay, organophosphates come from pesticides along with the war gases. So dichlorvos, malathion, parathion, but importantly serine and tabun. The V agents, that's also what they're called, victory, venomous, and viscous. They irreversibly inhibit acetylcholine esterase. And is this tonic paralysis? No, it's actually flaccid paralysis. The nerve impulses are continuous, muscle contraction and glandular secretions don't stop, so people froth at the mouth. That's one of the signs of organophosphate poisoning. And it's, though there's like the continuous muscle contraction, it's not tonic paralysis because the neurotransmitter will be used up, so eventually it just leads to flaccid paralysis. Like I said, eventually the receptors are unresponsive. Okay, so in myasthenia gravis, antibodies will bind to the nicotinic receptors, and that blocks just the binding of ACH to the receptor. It's less capable of exciting the postsynaptic muscle cells, and eventually you run out of acetylcholine. So you treat this with acetylcholine esterase inhibitor. So we don't want the acetylcholine to be taken away. We want to leave it in the junction so it can actually bind. And there's progressive weakness in myasthenia gravis, especially after like movement and whatever, because each time the person moves, there's just a decrease of acetylcholine because more is going to be released, but it takes so many acetylcholine to bind to the nicotinic receptors. It's competitive inhibition, right? So in Lambton-Eaton, it's going to affect the L type of calcium channels in the presynaptic terminal. So without the calcium allowing influx, it's not going to allow the acetylcholine to be released at all. So it's also caused by small cell lung cancer. So that will also like affect the receptors or the voltage gated calcium channels. So myasthenia gravis, there are the receptors affected, and Lambton-Eaton is going to be the calcium channels. Okay, toxins. Botox will enter the nicotinic cholinergic motor nerve terminal, and that blocks the release of acetylcholine. So it cleaves the presynaptic membrane protein involved in neuroexocytosis, causes flaccid paralysis. So it's similar to Lambton-Eaton, not in the same mechanism, of course, but kind of the same principle of blocking the release of acetylcholine. Tetanus is going to bind to the synaptic terminal, and it's going to increase the acetylcholine release. So it's going to be an overstimulation of acetylcholine, and it's going to cause spastic paralysis because it binds irreversibly to the receptors, and it forces the vesicle membrane proteins to continue in the neuroexocytosis. So there's going to be increase in acetylcholine release, and eventually it just blocks the neurotransmission. But the body still produces the acetylcholine, right? So this doesn't have anything to do with the other, like, with the organophosphates. So this person, it's not going to be able to make the acetylcholine, right? So they're going to run out. In a person with tetanus, they're going to continuously make it. So there's going to be like contracture. That's what it's called. because there's just going to be constant acetylcholine binding. Like I said, there's just an increase in acetylcholine release and eventually blocks the neurotransmitter. Catecholamines come from the tyrosine. Tyrosine is made in the pars compacta of the substantia nigra, and it's going to make dopamine thanks to the enzyme tyrosine hydrolase. And dopa, it's the precursor to dopamine. It's going to be stored until dopamine B hydroxylase catalyzes it, hydrolyzes the dopa, and then it forms norepinephrine in the postganglionic sympathetic neurons. So the noradrenergic or norepinephrine neurons are going to be in the locus coeruleus and the lateral tegmentum and reticular formation, and they're stored there until it's needed. Enzyme degradation is catalyzed by MAO-A, B, COMT, and PST. COMT is going to be the soluble form of SCOMT in the astrocytes. COMT is going to be formed in the neurons. And what does COMT do? It will break down dopa in the prefrontal cortex, and it's going to form phenylsulfotransferase it's going to be catalyzed by phenylsulfotransferase in the neurons. Okay, so what does the catecholamines do? It increases heart rate, of course. It's going to increase peripheral resistance and venous return. It's going to relax the smooth muscle, help contraction, and modulate the amount of fluid and electrolytes. It plays a role in glycogenolysis and lipolysis, and decrease the amount of sodium produced and the glomerular filtration, and it's going to increase aldosterone. So it's going to increase the amount of sodium reabsorbed in the body. Modulates the responses of a bunch of endocrine systems, and increase the secretion of glucagon and insulin. So in the mesocortical area, it plays a role in cognition, memory, learning, emotional behavior, and attention. In the nigrostriatal area, it plays a role in movement and sensory stimuli. And in the mesolimbic, it's going to be playing a role in pleasure, reward, addiction, emotion, and perception. Okay, so dopamine. Tyrosine is its precursor. It's made in the substantia nigra, along with the pars compacta, ventral midbrain tegmental area, and the arcuate nucleus. Controls the flow of information, movement, appetite, working memory, reward, pleasure, motor function, compulsion, and perseverance. So it has a lot of roles besides just, reward and pleasure. And it's any dysfunction, any kind of problems with the amount of dopamine is going to result in Alzheimer, ADHD, Parkinson's, Tourette's, schizophrenia, and addiction disorders, not just plain addiction. Okay, norepinephrine is going to be made in the postganglionic sympathetic neurons in the locus coeruleus, the lateral tegmental field, the reticular formation, and the one place outside the brain, the adrenochromaffin cells. Alpha and beta receptors are going to be G protein coupled. They use IP3 as its signaling molecule. It plays a role in recall memory, attention, and motivation. So hopefully you're paying attention to this lecture. Hopefully your norepinephrine is kind of kicking in. There is a monoamine hypothesis where serotonin regulates all the other hormones, and decreased serotonin will decrease the amount of norepinephrine, causing depression. And that's why we use that theory to prescribe SSRIs or norepinephrine and dopa reuptake inhibitors to treat depression. Serotonin, most of the serotonin is made in the GI, where it regulates just the bowel function and movement, reduces appetite and nausea, And the thing is, serotonin cannot cross the blood-brain barrier. It's not lipid soluble. So just because you eat a lot, that doesn't mean that you're happy due to the serotonin in your gut. It's serotonin in the brain. In the brain, it impacts mood, happiness, and anxiety, and it's made by like other structures in the brain, not from the gut. So if you read like a random Facebook post that says you can eat your way to like health and eat your way out of depression, that's not true. That would lead to like an addiction to food, and that's totally different. Then you'd be playing around with dopamine, right? Something that affects addiction. Okay, so in the blood, it also floats around, and it helps form the blood clots, and it's released by platelets. So that's it for now. Thank you for your attention. Thank you for hanging on after all of those tangents, but thank you once again, and good luck studying.